Hello? All right. I think he can hear my big mouth all the way over here. Hello? Hello? He can hear over there? Hello? Yeah, he can hear. Okay, so what chapter is this? Chapter 8. So today we're going to talk about cell cycle. How do cells divide? Which is that process of mitosis, right? So how do we create new cells, clones of our already existing cells for cellular repair, etc. And then we're also going to talk about the process of mitosis, which is the cellular division process only for your gamete cells, so only for your egg cells and your sperm cells. I threw these in first just to kind of see if you guys remember from our previous chapter. So these practice questions says DNA is found in a structure called centrosomes, chiasma, histones, chromosomes, or centromeres. Chromosomes, right? The other vocabulary word we've never even talked about, but we will today. The second question says the chromosomes of eukaryotic cells are found where? In the nucleus, right? So we're going to talk more specifically about what a chromosome is today. How does it duplicate? How do we separate it and replicate it? Well, not replicate it, just separate it. And we're also going to zone in here on the nucleus, particularly when we're dividing that genetic material. And of course, again, that process is called the process of mitosis or meiosis, depending on if it's an autosome or a gamete cell. All right, so how do we get from here to there, right? This process of cell division is continuity of life. So we must obviously reproduce our cells. Remember in chapter 1, we talked about one of the major characteristics of life to determine if you were alive. You obviously have to be able to reproduce, right? Reproduction can occur in unicellular organisms in a process called asexual reproduction, which we'll talk about when we talk about meiosis. We also use reproduction for growth, repair, renewal of cells. The cell cycle is the process by which we actually go in and separate this genetic material along with the organelles, the cytoplasm, and we go from one cell to two cells, right? Particularly in the process of mitosis, we're basically just creating a clone of the cell that already existed, right? Maybe some of your skin cells have scraped off. You need to renew your skin cells. Do you want to renew a skin cell that has a different genetic combination than previous skin cells? Yes or no? No, you want the same skin cell. Does it happen? It absolutely happens, right? Mutations occur.
Is that pigmentation?
Well, it could, yeah, depending on your melanin levels and determinations, but mainly I'm getting at what if there was some type of like UV radiation and you had some type of mutation in that skin cell, right? It can become cancerous, and we'll talk about that later on as well. But it's not a good thing if you're making cells that don't have the same genetic combination as your original cells in the process of mitosis. Not in meiosis, not in the gamete cells, but when we're growing, when we're repairing, when we're renewing old damaged cells, we want to produce the same cell. All right, so what is passed on to our daughter cells? We've talked about DNA, we've talked about the nucleus. You can see down here, your chromosomes are actually stained orange right here, right? And the green portion right here is the staining of your cytoskeleton. So you guys already know that genetic material is DNA, right? Deoxynucleic acids. The division step of that genetic material is called mitosis, which we'll talk about in detail today. So we actually separate that genetic material. But you also have to assort and separate the organelles and the cytoplasm. And so that portion of division is called cytokinesis, right? And we'll look at both of these in greater detail today. So duplicating our DNA, this is a process that is extremely complicated, and we actually cover DNA replication in future chapters. I think it's in chapter 10. Today we're not really going to be concerned, we're not going to talk specifically this process of how do we go from one strand to two strands. This process right here requires lots of enzymes, there's a lot of terminology in there, and again it's covered in its own chapter. We're going to really focus on how do we separate that material into two new cells, right? Not the actual process of duplicating the material. In DNA replication, we do split into two new daughter cells. The human cell duplicates, it's about 3 meters of DNA, so it's quite a large amount to fit in such a microscopic area. We separate the two copies, so each daughter cell has a complete identical copy. Remember, we're kind of making clones of ourselves there. And the error rate is about 1 per 100 million bases. There's about 3 billion base pairs in the million genome. So that's about 30 errors per cell cycle. Not a big deal, right? I mean, you're going to have mistakes when you have that many replications. Our cells have the innate ability in order to recognize these mistakes, hopefully, right? If they don't recognize the mistakes, is when we can have the cell cycle out of control, whacked, just replicating randomly, and that is called what? Cancer, right? Or a tumor can eventually form. So it's important that we keep that cell cycle in check. Notice there's also mutations, it says here, 30 errors per cell cycle. Mutation is not a bad word, guys. It doesn't always mean that it's going to have a detrimental effect on the cell or on the phenotype. Doesn't mean it's going to lead to cancer all the time. Most of our mutations are what we call silent mutations, and they actually occur in areas where there are no proteins encoded. We're going to talk very specifically about this when we start talking about transcription and translation of DNA, right? And we'll talk specifically about how mutations occur and what happens if they're detrimental or not. But for today, just know that they do occur during this replication process. DNA, hey. Yeah, we're going to use the, we want to see the, do you have the slides? Yeah. Of like the onion in the blastula? Yeah. I wanted to use the microscope again. Okay. So we'll be in there. All we're going to do is view the slides. Okay. Thank you. All right, so DNA is organized into this structure called chromosomes, right? So the double helix of your DNA molecule. The associated proteins of this chromosome is called histones, and the DNA protein complex is called chromatin. So just a lot of vocabulary words here, but they're all found in the nucleus. If you look here, here's your double helix, right? We've talked about the structure of that double helix. And guys, we just said that the DNA molecule can go up to 3 meters in length. So how do you fit that into this little bitty tiny area called the nucleus? And the answer is, you know how you, let's say if you hold a rubber band on each side, and you hold it on one side, but you begin to twirl it on the other side, what happens? The rubber band starts to like coil up back upon itself. Does everybody agree? Yes? So that's exactly what DNA does. But it coils back upon itself with the aid of these histone proteins. So these little circles right here, these are histones. They're proteins, and they allow the DNA to kind of coil up so it's nice and compact in the nucleus, which is going to be very important, particularly when we're about to go through cell division.
3 meters long in each nucleus, or all together throughout your body?
Well, in each cell, right? And so there's a cell, I mean, yeah, there's billions of cells. So yeah, in each nucleus.
Okay.
And then when we have this supercoiled structure, you can see it's wrapped around the histones. This supercoiled structure causes the chromosomes to look like this X kind of, right? And you may have seen this X like on TV or newspaper articles and stuff. This is a unique structure. When the DNA is actually coiled up like this, very tight, and it looks like a little X, the chromosome itself, this means we're about to divide. We're about to go through the cell cycle, through cellular division. Chromosomes or DNA doesn't always look like this, and we'll look at some differences, how I can tell if a cell's about to divide or if it's not, based on its structure and its tight coiling with those histones. So after we duplicate the DNA, as we just said, the chromatin will condense, right? So after we've copied it, it condenses into this X-like chromosome structure. The coiling and folding make it a smaller packaging system. And from DNA to chromatin to highly condensed mitotic chromosomes right here. And you'll see, guys, you guys see there's 2 copies? Yeah, we'll talk about that in a minute. But I had 1 copy. When I replicated it, I had a 2nd copy. This is a complete chromosome. These 2 copies are called sister chromatids, same exact genetic information. How we went from 1 copy to 2 copies is the process of DNA replication, which we talk about in chapter 10. But for now, just know, the sequence of A, T, G, and C's in this copy is exactly the same as the sequence of A, T, G, and C's in this copy. Sister chromatids, they're held together right here in the middle by something called a centromere. This is going to be really important because now that it's nice and coiled up and we can clearly tell where the 2 copies are, it's going to be much easier for that genetic information to separate correctly into 2 new cells. Everyone agree? If it was just this big mess ball, all this looks like a big old mess of yarn, it'd be really hard for the cell to decipher which is which and separate it. So this condensing right here also makes that cell division process more efficient and ensures that each new daughter cell is going to get the exact copy of DNA, or the correct genetic material. Definition of a chromosome. Again, when we duplicate the chromosome, it's still called a chromosome, whether it's one copy or two copies, right? So here you can see, here is before the chromosome has been replicated. After the chromosome has been replicated, which just means an exact copy has been made from it, one chromosome, this is still one chromosome. It just has two copies. It has double the amount of genetic information. Does everyone agree? Yes or no? If I have 10 micrograms of DNA here, it means I have 20 micrograms of DNA here. Yes? I just double the quantity, the amount. Again, the middle portion here that holds those two sister chromatids together is called a centromere, and this is an actual transition electron microscope of a chromosome. So I've duplicated it. The process of now separating this new genetic information into two new daughter cells, that's the process of mitosis. This right here is what we're going to talk about today, right? So one chromosome, two chromosomes. Does everybody see down here? Once I've separated it, I now have gone from 1 to 2 because I've released that centromere and I put it into two new cells, or two new daughter cells, right? They contain identical copies of the chromosome's DNA, very important, right? So all your daughter cells are going to have the same genetic information that the parent cell had. Unless, of course, there's a mutation or some problem. All right, a duplicated chromosome consists of two centromeres, centrosomes, genomes, sister chromatids, or genes. Sister chromatids. Same genetic information? Yes. Sister chromatids are joined at the chromatin, the centromere, the spindle, the centrosome, or the centriole. Centromere, right? So be careful with your vocabulary in this chapter. A lot of these words sound the same, right? But they mean different things. All right, so let's take a look at the big picture here of the cell cycle. So down here, you can see there's a bunch of different parts here. The first thing you should notice is that I have this long phase called interphase. Everybody see that? Yeah? Interphase? Interphase is 90% of the cell cycle. This is where the cell spends most of its life. Then I have this little bitty phase right here called mitosis, right? This is where it actually, we separate that genetic information. And then remember, cytokinesis was when we separate everything else. So the cell spends most of its life in interphase. And then you should also notice that interphase is actually made up of 3 subphases. G1, the S phase, and G2. Yes? Does everybody see that? In interphase, the characteristics of interphase is the nucleus is well defined, so you can see the outside of the nucleus, just like you guys did when you looked under the microscope, and you'll see it today. And the DNA is loosely packed into long chromatin fibers. So it's just kind of hanging out. We'll look at each one of those phases in just a minute. Here's another diagram. I put this one in here because I like that it actually shows you the quantity of the DNA. So again, here's my interphase. The cell's hanging out. We separate it, and then we've separated everything else. Each one of these phases, it's on the future slides, but I'll just tell you right now. The G1 phase is called the gap 1 phase. And this is where the cell just hangs out. He eats, he released waste, he does his everyday job, whatever he's supposed to do, if he's a heart cell or a secretion cell, or it doesn't matter, right? The S phase up there, which stands for the synthesis phase, what happens? I have 1 copy, I have 1 copy. At the end of the S phase, how many copies do I have? 2. This is where DNA replication occurs, right? I go from 1 copy to 2 copies. Again, chapter 10, we'll talk about how that works. But right now, just notice I've doubled the amount of DNA that I have, right? I now have sister chromatids, same exact genetic information on each one of those arms. In the G2 phase, does my chromosome number change, quantity change? Nope. The G2 phase is the cell is preparing to divide. He's preparing to enter into mitosis. So he's making extra organelles, extra cytoplasm, extra plasma membrane. He's growing larger and larger because he knows he's about to have to divide and share everything between two daughter cells, right? I now enter into mitosis, or the M phase. Where did I go? I had one chromosome here. At the end of mitosis, I have two new daughter cells with one chromosome. I've just separated that genetic information. And then cytokinesis, which is right here, is the actual separating of the organelles and all the other cytoplasm. So this is what I started with. This is what I ended with. Did the chromosome number change? No, chromosome number didn't change. You ended up with the same exact cell, right? I started with one cell. I ended up with how many cells? Two cells, right? Everybody agree? With the same genetic information, same chromosome number. Yes, Maida, you had a question?
So you have two daughter cells, but one chromosome?
You have two daughter cells. Each daughter cell has one chromosome in it. You actually, so in totality, Maida, you've actually have two chromosomes. They're just in different cells now. Does that make sense? Yes? So one, right? But total two, because you have one in each cell. So you end up with exactly what you started with. You've just made a clone of that cell. All right, big picture. So, you can't really see that very well. But I encourage you to click on this animation. It's just it's lighted out because I already clicked on it. It's a great animation, and again, this is a process. So I'm not going to look at it right now, but if I were you and you're having trouble kind of going through this cycle, I would open it and make sure you review it. Okay, so here are the phases again of interphase. As I've already said, G1 is the first gap. Cell's doing his job. The cell's growing. And it's just flipped here. So where is he? Here he is. G1, right, of interphase. The S phase, DNA replication occurs. You copy that chromosome. You go from 1 to 2 copies. In the G2, or the second gap phase, prepare for division. The cell grows a little more. He makes more organelles, proteins, membrane proteins, because he knows he's about to enter into the M phase, right? And this is the first time you'll see the M phase actually has subdivisions as well. It can be divided into prophase, metaphase, anaphase, and telophase. I just remember it as PMAT. The high school kids have like crazy acronyms and stuff. I forget what they are. But they go in that order. So there's, if you're able to look at this little diagram, make sure you know what's inclusive of what, and G1, S, and G2 is an interphase, and so on. All right, so now we're going to look at each one of these phases of mitosis, defining characteristics, and what's going on in each one of those phases, right? So interphase of G2, we're at the very end of interphase right there. The cell's been making organelles and proteins. The nucleus is still well defined, right? So I still have a nucleus at the end of that G2 phase. In these pictures up here that correspond with the phases, the purple portion is the DNA that's stained, and then the fluorescent yellow, green, whatever that is the cytoskeleton that is stained, or the centrioles, and that'll be important in the DNA dividing. You'll notice that the DNA is still loosely packed into long chromatin fibers. So does it look like that X structure, that condensed structure yet? No, not yet, right? But it's about to. It's just slowly starting to get itself together. When you look at the nucleus, it still just looks like a big mass of purple stuff everywhere. You can't actually see the strands yet, right? In this stage, again, this guy is preparing for mitosis. He's making more proteins and more organelles. So on a slide, we'll look at some examples. This would just be like a really fat cell. He still has one nucleus, but he has lots of things going on inside of there, right? And so I can kind of tell the difference between a G1 and a G2 phase based on how big the nucleus is, right? Or how much genetic information is in there. Ooh, that's out of order. Which of the following occurs during interphase? So between that G1, that S, and that G2. Chromatin becomes tightly coiled. The chromosomes duplicate. Sister chromatids separate. The mitotic spindle forms, or cytokinesis occurs. B. Chromosomes duplication. Now, Samantha, you said sister chromatids separate, and they do, but not in interphase. They're going to separate during mitosis, right? Remember,
Oh.
interphase was that long phase, 90% of the cell, the G1, the S, and the G2. So B occurs during interphase. A, C, D, and E all occur during mitosis, right? And we'll point out where those occur when we look at mitosis closer. So here again are your phases: PMAT, prophase, metaphase, anaphase, telophase, right? And it's basically the division of our genetic information, followed by cytokinesis, division of everything else. God, that thing is so loud, isn't it? Okay, I love this picture. So this, we fluorescently stained the genetic material of a cell. So you can't see anything else. It's all blacked out. The only thing that's stained is, are the chromosomes. So look down here, guys. Notice here, this is the cell that I start with. That's the cells that I end up with, right? This is the, well, no, it isn't the cell I start with, sorry. This right here is starting in the S phase, yes? After I've duplicated my DNA. So notice the quantity. I have one nucleus. I have two nuclei there, but the DNA, do you see how the genetic information is large, right? It looks like 2 times as much as over there, yes? Do you guys see that?
Yeah.
So this cell is really large. He's probably in the G2 phase. He's already had his DNA replicated, and he is now entering into prophase. See how the DNA is starting to coil up? Yes? Prophase, DNA is coiling up. Remember, the DNA starts to coil up and become tightly coiled because it's much easier to separate that DNA, right? He's entered into prophase. They're now coming together, all the chromosomes. They're all wiggling around. They're starting to line up, and it's kind of hard to see right between here and here, but they've actually lined up in the middle of both of the cells now, right? Or in the middle of the cell. It hasn't actually separated yet. Then they begin that genetic material. Right here, those sister chromatids break apart, and they begin to move towards opposite ends of the cell. Does everybody see that? There's still one cell right here, right? One cell, but the genetic material is just moving to opposite ends. Once the genetic material is moved to opposite ends, cytokinesis begins. Now, telophase and cytokinesis kind of overlap each other a little bit. You know, you're separating organelles and cytoplasm at the same time you're separating your genetic information. Cytokinesis, and at the end you end up with 2 daughter cells, right, with genetic information. And notice, is the chromosomes coiled right now? No. What phase are these in? Interphase, and particularly what phase? G1. Everybody agree? Yes? G1? Then you go through S phase and you'd end up here again, right? So this division of genetic information, this shows a nice turn of events, that doesn't sound right. A nice flow of how the genetic information is coiled up, lines up, separates, and then eventually is put into 2 new daughter cells. Samantha, did you have a question?
Yeah, how come our starting cell is an interphase?
Well,
Because wouldn't it be moving to G1 and then...
so we just skipped this part. There's a spot down here. Remember, interphase is made up of G1, S, and G2.
Yeah.
So this is G1. We just skipped the S phase right here, where it duplicated.
Oh, I get it.
G2.
Okay.
We just didn't. Down here, it went, the DNA, this cell, like one of these cells, came down here and duplicated his DNA and looks like this.
Okay.
Right? So we just
Yeah.
kind of started with G2
Okay.
in this picture. All right, the cell cycle results in the production of 4 cells. Does it produce 4 cells or 2?
2.
2. So I can eliminate A. 2 cells, each with the same amount of genetic material, but with different genetic information.
No.
2 cells, each with the same amount of genetic material and the same genetic information.
Yes.
Yes, best answer, right? And if you were to read over the other ones, 4 cells, we know it's automatically not right, and different amounts, and that's not right. So best answer is C. All right, so let's look at each one of these M phases. The first one is prophase, defining characteristics. The chromosomes are now visible. They've coiled up. You can see them up here. It looks like a bunch of spaghetti, right? They're beginning to coil. The fibers extend from the centromeres. So here's your centromere. These fibers, the early mitotic spindle, actually begins to form. This is part of your cytoskeleton that begin to move to opposite sides. Those fibers are going to be really important in order to help move the chromosomes to opposite sides of the cell. Again, the centrioles move to opposite sides of the poles. So here are your centrioles. They're beginning to move to opposite sides to create two sides of the cell. Fibers form, mitotic spindle, as we just said. Actin and myosin are just part of the cytoskeleton. So, I don't know if actin and actin is just like the same fiber that you would use for muscle contractions. That'll be important later on. The nucleolus begins to disappear. Nucleolus was this densely stained area in the middle of the nucleus, which was responsible for making ribosomal RNA. And the nuclear membrane begins to break down. So it's not completely gone yet. Remember, the nucleus is that double membrane that houses the DNA? The nuclear membrane out here just slowly begins to break down, right? Eventually it's going to have to disappear, so that way you can separate your genetic information. Proteins are also attached to something called the kinetochore, right? And so the kinetochore is actually what keeps that together. Microtubules begin to attach to the kinetochore, and the chromosomes begin moving. So when you look at that animation, they kind of like start wiggling back and forth. They become really uncomfortable and start moving towards the middle. You can see up here, this is still part of prophase. Here are those stained centrioles. The spindle fibers are beginning to attach themselves to the centromere right here of the chromosome. So that's important. There's an actual attachment here between the spindle apparatus, or the spindle fibers, and the chromosome. Where the spindle fiber and the chromosome actually attach to each other is called a kinetochore, right? And the attachment of this is going to aid in the pulling of the chromosomes to opposite sides. So it's just like a little mini motor. Here you can see a close-up of the kinetochore. So this is a chromosome. Centromere is right here, all right, the part that holds the two chromosomes together. The kinetochore is actually a protein that's going to be attached here to the spindle apparatus, right? And so, guys, as soon as this association breaks is when you enter into a new phase. As long as this is still together, right, we haven't separated into two new cells yet, right? It's a protein complex that holds it together. All right, the next phase is metaphase. M for middle. Meta means middle. You line up at the middle. Defining characteristic of metaphase are the chromosomes will line up in the middle of the cell. This helps to ensure that they will properly separate, right? That each new sister chromatid will go to a new daughter cell. Of course, this would be the perfect picture. You can see they're lining up. It's kind of hard to see metaphase, you know, as long as you see a big clump in the middle of it. But you can see they're obviously right here in the middle. And you can see that those spindle fibers and those centrioles are clearly defined right now, right? You can clearly see that they're attached to the chromosome. The second this centromere breaks and the kinetochore association is broken between the two sister chromatids is when you enter into the next phase of mitosis, which is anaphase. So during anaphase, the sister chromatids begin to move to opposite ends. Clearly you can see up here, they're moving. You can see here what happened. Those two sister chromatids were broken apart, and one sister chromatid is being pulled to this side, the other one's being pulled to that side through the spindle apparatus. There's ATP being used right here, right? It's like winding it back up to the other side, right? To each side of the cell. They're pulled at the centromeres. Pulled by motor proteins. It's like walking along those microtubules, right, to get it to the opposite side. During this time, you usually see an increased production of ATP by the mitochondria when cells have to divide. And the poles also begin to move farther apart to lengthen the cell, so it becomes more obvious that there's going to be two new daughter cells, right? Instead of that round structure, it becomes kind of that barbell structure. The separation of the chromatids, what I just said. So this is one chromosome, two sister chromatids. The second that becomes separated, you now have two chromosomes, right, which are moving to opposite ends of the pole. So those kinetochores are very important. A signal is produced when they break and they begin to move to the opposite side. So that's defining again when we go from metaphase to anaphase, as soon as they break and start moving to opposite sides. And the last phase of mitosis is telophase. The chromosomes actually arrive at opposite ends of the pole. The daughter nuclei form, the nuclei begin to reform, the nuclear membrane is now building itself back up again. You can see here the genetic information is clearly on opposite sides. You probably have some separation of cytoplasm. Cytokinesis is starting kind of at the same time, you know. There's no real defining characteristic there. The chromosomes begin to disperse. You can no longer see them under the light microscope, so they're not condensed anymore. They don't have to be. They've already separated. So you can see they don't have that coiled form anymore up there or in the cartoon. The spindle fibers disperse. They disappear. They're no longer needed. And cytokinesis begins, the actual division of everything else in the cell besides the genetic information, right? So they start to share plasma membrane, proteins, enzymes, organelles, cytoplasm. Cytokinesis is obviously the last. It's not actually part of mitosis, right? But it's the phase that follows it. This cleavage furrow forms, and you can see here, see how there's still some cytoskeleton left over here? This is actin, and you can still see it right here as well. Remember I told you those are the same fibers that are responsible for muscle contraction? So it's like one big muscle contraction that occurs between two animal cells, what we call a cleavage furrow, and it like pinches the two new daughter cells off so they can actually separate completely from each other. So it says it tightens to form this furrow, which splits the cell into two. It's like tightening a drawstring, right? Pinches it off. And on the right-hand side over here, this is an actual electron microscope view. You can see the cleavage furrow that's forming right here. Can this happen with a plant cell? What does a plant cell have on the outside of it that animal cells don't have, and it's really thick? Cell wall. There's no pinching going on in the cell wall, right? It's way too, it's thick, thick outer cell wall. You can't, it's not very flexible. Remember, it's not fluid. We don't have this fluid mosaic model here. So plant cells do something a little differently. Oh, we'll look at that in just a minute. Let me show you, I want to show you mitosis in a whitefish flagellate. So this is an animal cell. You're going to view this in the microscope today as well, where the cleavage furrow forms. Interphase. I know it's interphase. I can tell just because it's not coiled up, right? I mean, the chromosomes aren't coiled. Early prophase, chromosomes begin to coil. Metaphase, defining characteristics, the chromosomes are in the middle. You can also see the two spindle, the centriole spindle apparatus clearly is formed. Anaphase, remember I told you, this one's a little, if you were actually like zoom in on it, it would be the second that those two sister chromatids are moving to opposite sides, right? They've broken here. Early telophase, genetic material still being separated. Late telophase and cytokinesis at the same time. The genetic material has been completely separated, and you can see the chromosomes are beginning to uncoil, right? And then eventually that cleavage furrow would pinch off, and you would have two new daughter cells. These are the diagrams that you're reviewing in the PowerPoint itself. Again, you know, if you can identify defining characteristics in your head as a picture, it's much easier to remember, right? So anaphase, this one's easy to see that they're separated. I can see the difference here, right? Coiled, not coiled yet. Late telophase, and you can even see the DNA quantity. So look, I mean, this is a lot of DNA, right? Now, this is about half the amount of DNA. So I know that I've successfully separated that DNA. And here is that cleavage furrow right here acting to like pinch off between those two cells, right? Eventually it'll pinch off that plasma membrane. However, as I said, or as you guys said, plant cells have thick outer cell walls. They cannot form cleavage furrows because of that outer cell wall. So the plant cells do it a little differently. They actually begin to import these vesicles, which are derived from the Golgi right here in between the two cells. And eventually all those vesicles fuse together to form a new cell plate between the two cells, right? So just a little different process. You can see here's an actual close-up of a plant cell. And you can actually see here the little vesicles, right, that are being imported from the Golgi. Eventually they'll all fuse together to form a cell wall just like this, right? That's on the outside of the plant cells. But everything else before that is exactly the same in a plant cell. The mitosis and prophase and metaphase. Samantha?
Does our process happen quicker than a plant cells would? Is that why, like, if we get a scratch, it would maybe take a day or two for it to heal, whereas if a plant were to get, like, cut or scratch, it would take a
It depends.
longer
It
to
would.
go over?
Yeah. And it depends on where it is. Remember, like mouth saliva cells, right? Tongue cells heal much quicker than maybe like a cut that you got on your toe. Depends on the type of cell, where it's located, how bad the injury is. Guys, it also depends on your immune system, right? I mean, someone that has a really strong immune system is going to heal much faster and produce new cells faster than someone that has a very weakened immune system. So the answer is yes and no. There's so many factors that determine how fast this process occurs. All right, so mitosis in plant cells. Again, you can see all these phases look exactly the same. The only difference is in telophase, you're not going to have that cleavage furrow, and it's kind of hard to see right here, but this stain is actually the cell wall, and it's beginning to form and import those vesicles. But the prophase and the metaphase, it all looks the same, right? It's a little square, right, because of the cell wall, but the actual separation of the genetic material occurs exactly or in the same way. All right, here's a picture. You should be able to identify, under the microscope you'll see them, but also for your exam, you should be able to pick out the following cells for me. So, the first thing is, I'm just going to make some inferences here. This guy is probably in a G2 phase. Does everybody agree? This guy right here? Chromosomes aren't coiled, so I know he's not in mitosis. But he's really large. Does everybody see how big he is compared to these two? So these are probably in G1. This guy's probably in G2. Everybody agree? Yes, because they haven't actually doubled my DNA quantity yet. The chromosomes are beginning to coil. So D is probably in what phase?
Prophase.
Probably in prophase. Agreed? Most likely in prophase. Haven't separated anything yet. He's still really large, but the chromosomes are beginning to coil up, which is a defining characteristic of prophase. This guy is probably in which phase? Metaphase. Everybody agree? Lines up at the middle. M, meta, middle. This guy, B, is probably in what? Anaphase. Moving to opposite sides. Anaphase, early telophase. And this guy is probably in telophase, right? And or, he said cytokinesis, so you can't really tell, but they kind of overlap. So make sure I could ask you the same questions I just asked you on the test. See? Yeah, that's all you need to know. Okay. Okey-doke. Practice. Which of the following occurs during prophase? The chromosomes line up in the middle. The nuclear envelope forms, sister chromatids separate, the mitotic spindle begins to form, or cytokinesis occurs. A, B, C, D, or E? D. Everyone agree? Mitotic spindle begins to form. In what phase do chromosomes line up at the middle? Metaphase. The nuclear envelope begins to reform again during what? Telophase. Yes? Sister chromatids actually separate during which phase? Anaphase, right? Yes, agreed?
There's so many phases.
Sister chromatids form or DNA replicates during which phase, Chris? The S phase of interphase. Is that what you were thinking?
Yeah.
The duplication of the DNA, where you do have sister chromatids in interphase, right, during the S phase. And cytokinesis occurs, well, following the M phase, right, or following telophase. Immediately following telophase are kind of like, they overlap a little bit. But the best answer to the question is D. Which of the following is a stage of mitosis? Cytokinesis, telophase, meiosis, interphase, or DNA synthesis? Which one of these? There's only one that's actually part of the M phase. And it is B, telophase. Agreed? None of these are meiosis, we haven't even talked about. Interphase is not part of the DNA synthesis is part of interphase, in the S phase particularly, right? So, and cytokinesis is not part of mitosis. It's a stage that follows it. So, the answer is B, right, is part of mitosis, or the M phase. The correct sequence of stages of mitosis are A, B, C, D, or E. the answer is A. During metaphase, nuclear envelope breaks, cytokinesis occurs, sister chromatids move, centromeres divide, or chromosomes line up in the middle. E. Yes? A, B, C, D, E. E, middle. Which of these events occurs during anaphase? Y'all, I'm not going to read it again. A, sister chromatids separate. A cell that completed the cell cycle without undergoing cytokinesis would have less genetic material than it started with, not have completed anaphase, have its chromosomes lined up in the middle of the cell, be prokaryotic, or likely to have 2 nuclei.
Have 2 nuclei?
Yeah, have 2 nuclei, right? If you don't actually go through that final part where you separate, you're probably going to be 1 cell with 2 nuclei. It
What
happens.
happens to that cell?
Hopefully the cell cycle, the trigger factors are in there and the cell dies. I mean, hopefully he doesn't stay alive.
But would that be the form of cancer if that cell were to stick around?
No. We'll talk about cancer, yeah. No, so guys, your cells have internal mechanisms of saying like, something's wrong with this cell. We have to just, it's called apoptosis. You actually get rid of it yourself. Yeah, and your body does it all the time. It's a pretty fascinating, when those mechanisms, or what we call those checkpoints, begin to not work anymore, is when things like cancer start to develop, or you start seeing mutations that you can see phenotypes of, or you get skin cancer, or things like that happen. So remember, those errors happen all the time. I mean, 30 per cell cycle, so it's not a big deal. If you have an error, your body fixes it. It's when your body stops fixing it. It's when you can run into problems. So this cell would not likely survive, right? It's just like, if you produce an egg cell that's, missing a few chromosomes, that cell is not likely to survive. Every once in a while it does, right? And you can have, I don't know, premature baby, or, malformations, or whatever it may be. But more than likely, the cell is going to be eliminated by your body. And here's what we just said, right? So we're going to talk more about cancer in the next few chapters when you guys actually start to understand what transcription and translation is. But all cancer is, in simple terms, is the checkpoints within the cell cycle that we just discussed are out of whack. So an error occurred, the checkpoint did not kill the cell or stop the cell from replicating any further, and so he just keeps replicating and replicating and replicating and replicating and replicating until you get this tumor. Eventually, if the tumor metastasizes or begins to move into the bloodstream, the tumor cells can then spread to lymph nodes and other parts of the body, right? So all cancer is, in simple terms, is your cell cycle that's out of control. It's not regulated anymore, right, in a particular cell, those checkpoints. I use this as my Henrietta Lacks. These are your HeLa cells, or HeLa cells. These are the first cells, well, just watch it. We'll talk about it next time. It's a good story. All right, meiosis and sexual reproduction. So we've talked about the cell cycle and mitosis. If we want to just make new skin cells or heart cells or whatever types of cells we need. The next process we're going to briefly talk about is called meiosis. And it has a lot of things in common with the process of mitosis. There's a few differences, which are major. And we only use this process of meiosis in sexual reproduction. We only use this process to make sperm, if you are a male, and eggs, if you are a female, right? So it's a different process used for only eggs and female cells, gamete cells. Mitosis is used for every other cell in the body, known as an autosome, right? Everything else uses the process of mitosis. So mitosis, just to review, produces cells with the same genetic information. You're making clones of your cells, same amount of DNA as we just saw, same number of chromosomes are made in the division, same genetic information. There's no recombination. Hopefully it's the exact same genetic information, with the exception of a mutation. Asexual reproduction, on the other hand, these are single-celled eukaryotes, which you can reproduce asexually, such as yeast, paramecium, amoeba. You can see up here, this is a hydra. He actually just starts to bud off and make his own offspring. So it'd be like if I could just make my own babies, right? I wouldn't need a sperm cell. As you can imagine, that's just like creating a clone of yourself, right? So in asexual reproduction, the information is exactly the same in the offspring as it is in the parent. Yes? Everybody agree? So it's just kind of like mitosis, right? You're just producing clones of yourself. These are very simple organisms, single-celled organisms. Simple multicellular organisms, again, Hydra would be an example of a budding yeast. It says, what are some advantages of asexual reproduction and disadvantages? Well, advantages of this, it happens really fast, and you can produce lots of babies really, really fast, right? That's a good advantage. Very small genome, you can multiply fast. Whereas us, it takes 9 months, right, in order to produce a baby. Disadvantages is just when you think about inbreeding, particularly of dogs or of human beings, you don't have a way to breed out the genetic material that isn't an advantage to you, right, or to an organism or to your offspring. So remember that process of natural selection we talked about, right? So the more that you breed with throughout your population, right, not in your population, you're more likely eventually statistically to breed out those bad traits, right, or whatever those mutation traits are. So the less sexual variation that you have, the less genetic variation that you have, the more likely that those bad genes or mutations or undesirable characteristics are going to stay within a gene population, right, or within a gene pool. Here's budding yeast. Binary fission in amoeba, again, they replicate really fast. They just make little clones of themselves. Same genetic information. But what about the rest of us, right? So we make egg and sperm. What if we made an egg cell and a sperm cell? And by the way, we have 46 chromosomes, right, in each one of our cells. If I had 46 chromosomes, so all of the cells and all the other body, they all have 46 chromosomes. Everything has 46 chromosomes. If my egg cells had 46 chromosomes and my sperm cell had 46 chromosomes, when they came together, the zygote would have how many chromosomes? 92. Yes? So my baby would have double the amount of chromosomes that I have. And then her baby would have double the amount. Does everybody get it? Right? So some kind of way we have to reduce this in half. Right? We can't go through the process of mitosis because we would just like exponentially start increasing the number of chromosomes of every one of our offspring. That would be crazy. So only in egg and sperm cells, we have to cut this number in half some kind of way, right? So that way, upon fusion of an egg and a sperm cell, we get the normal 46, just like we have, right, or humans have. All right, so here's what we call a human female karyotype. If you count these up, there's 46. You have chromosomes 1 through 22, and then you have your 2 sex chromosomes down at the bottom. You'll notice that, okay, listen, this is like kind of confusing. So if you were to actually zoom in up here, this is actually 2 sister chromatids. It doesn't matter if there's 2 sister chromatids or not, right? It just means if you're in G1 or G2 phase. There are how many copies of chromosome 1? No. How many copies do you have of chromosome 1? 2. How many copies do you have of chromosome 7? 2. How many copies do you have of chromosome 14? 2. You have 2 copies of every one of these chromosomes because you get one from your mom from the egg, and you get one from your dad from the sperm. Everybody agree? Yes? Agreed? Are they the same genetic information? No. No, they're different because one's from your mom, one's from your dad. These 2 chromosomes are called sister chromatids. Same genetic information. These 2 chromosomes are called homologous chromosomes. Homo means the same. Everyone agree? Yes? They are the same, they're chromosome number 1. They're the same chromosome, homologous. One came from your mom, one came from your dad. The sequence of A, T, G, and C's are going to be different, right? Because we have different genetic, sequences, but they're the same chromosome 1, right? So, I don't know, if this is the gene for eye color, well, this is the gene for eye color from your mom, this is the gene for eye color from your dad. Yes? So there's a difference between sister chromatids and homologous chromosomes. Doesn't matter in mitosis, it matters in meiosis. And then down here you'll notice if you are a female, you have 2 X chromosomes, right? And if you are a male, you have an X and a Y chromosome. Only difference. This is a nice karyotype. I wanted to show you this just so we could reinforce what I just said. So this is an actual karyotype, right? They just sequence it. And a lot of times they do karyotypes to test for things like Down syndrome, genetic disorders, if you're missing a chromosome or something. But if we were to blow chromosome number 1 up, again, this is a pair of sister chromatids. This is what we talked about in mitosis, right? Same genetic information, it went through the S phase. Both of these are homologous chromosomes, different genetic information. Everybody see that? One from your mom, one from your dad. In mitosis, it didn't matter. I didn't care about homologous chromosomes because everyone just separated exactly the same way. In meiosis, I care about them. In meiosis, homologous chromosomes actually find each other and they like pair up to exchange genetic information. Imagine if this is chromosome number 1 from your mom and this is chromosome number 1 from your dad, and they come really close together. They actually do come together, like they attach to each other, and they start like switching out genetic information. Does that happen? It does. It happens. It's the same. It's why, you know, I may have 25 daughters, but they're all going to look a little different. They're not going to look the same, right? Even though, I mean, I have the same genetic information coming from me. My husband has the same genetic information coming from him. Upon fusion, it's the same genetic information, but something special happens in meiosis. And these genes actually start to switch out genetic information, right? Which is why my sister may look kind of like me, but she's not going to look exactly like me, unless she's a twin, right? Does this happen in mitosis? No. This is one of the variables that contribute to genetic variation, right, among a population. All right, homologous chromosomes carry the same genes, include only autosomes. Again, autosomes are everything else except for gametes in your body. Are a set of chromosomes that the cell receives from one parent, include only sex chromosomes, or separate during interphase. Carry the same genes. They carry the same genes. They carry the same. Maybe you carry gene APXDG57. Same gene. The sequence of the gene might be different. Everyone agree? We carry the same. We both carry the eye color gene on chromosome 3. The sequence is going to be different. It's going to be expressed differently, right? They carry the same genes. What sex chromosomes belong to a normal human male? Anyone? XY, D, and of course we said female is XX. So how do we make egg and sperm? And as we said, we need to reduce that number in half, right? So through the process of meiosis, we actually go from 46 to 23 chromosomes. We actually cut that chromosome number in half, which we did not do in mitosis. So we must have some type of like double division, right? And we do. So that way upon fertilization, you can restore that chromosome number to 46. Only occurs, again, in your gamete cells. So here's what we call this alteration of generations, which means that we're going from haploid to diploid, haploid to diploid. So let me define for you quickly what's the difference between a diploid and a haploid cell. Di means what? 2. So just imagine a diploid cell probably has both copies, a mom and a dad copy. Haploid probably has how many copies? 1. So if I have just a normal cell hanging out, I'm a diploid. 2N just means you have 2 copies. It means you have a copy from your mom and a copy from your dad. Through the process of mitosis, they develop, and then there's your sex cells, your specific cells, then go through what we call a meiosis process. And I no longer have 2 copies. I have what? 1 copy. So haploid cells are gamete cells. They only have 23 chromosomes, which we just said, right? All the other cells have how many chromosomes? 46. So diploid is represented by 2N. Haploid is represented by N. In humans, we have to go from diploid to haploid to make gamete cells. Different process. Meiosis reduces this chromosome number in half. And upon fertilization, we restore it, right? So from haploid to diploid. This is a little confusing to people sometimes. Don't
Yeah,
like
I'm super confused
it's
by that.
yeah. Don't spend a lot of time on it. Just know I know because it took even my high school kids. It takes them forever. They don't get it sometimes. Let's go through the whole thing first, and then it might make a little more sense. The difference between diploid and haploid. But when you first hear it, it's really confusing. All right, big picture again. There's a wonderful animation here that you should watch to decipher the difference between meiosis and mitosis. Here's the big picture, right, of meiosis and how it's different. So meiosis also goes through interphase, just like mitosis, right? So the cells still go through G1, S, G2. They still duplicate their chromosomes. How many chromosomes do I have here?
Together, 92.
No, just in this one cell.
Oh, 46.
No, just in this one cell. This one. The one right here. How many chromosomes do I have? 2.
Oh,
Agreed?
okay,
Yeah? How many homologous pairs do I have in this cell? 1. Right? You have one from your mom, one from your dad. Homologous pair, everybody agree? That's a homologous pair? Yep. After I've gone through the S phase, my chromosomes have duplicated. How many chromosomes do I have here? 2. I still have 2 chromosomes here. But what's the difference? I just have double the quantity of DNA. Everyone agree? I have 2 chromosomes here. I have 2 chromosomes here. This has gone through the S phase. So these are called what? Sister chromatids. Same exact genetic information. I've just duplicated it, right? But in meiosis, homologous chromosomes like to stay together, right? They like to stick together because they want to make sure that they can find their partner to kind of switch up some genetic information. So interphase is exactly the same, right? We go from one copy, so you still have a copy, you've just duplicated it through the S phase. Samantha.
Okay, then what has 46 chromosomes?
A real cell. I don't want to put 46 on here because it would get too confusing.
Okay, but there's 46 in that cell,
There is,
right?
theoretically,
Okay,
yeah.
Cool.
We're just focusing
We're just
just
focusing
on one,
on-
just
okay.
so we can see the process easier. But yeah, I mean any other normal cell, a skin cell, a heart cell, a liver cell, blah, 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 everything has 46
Okay.
except gametes, right?
Okay.
But yeah, that's important to know. I'm just narrowing it down to 2 so I can show you what happens to a homologous pair. But same, right? You just have replicated that genetic information. There are 2 separate divisions in meiosis. The first division of meiosis, I actually separate what? What happened? From here to here, I've gone from 1 cell to 2 cells. Everyone agree? Yes? And what have I done? I've separated what? Watch your terminology. I've separated homologous chromosomes. Everyone agree? I've just separated the mom copy and the dad copy. Yes?
I guess that's why I'm a little confused. Why would the 2 cells want to separate when they're trying to mate?
Well, because they have to go through, so it's very important that they make sure that each new egg or sperm cell gets an individual copy of that genetic information.
Okay, now each of you, have they already shared genetic information?
Nope.
Okay.
Well, they have actually, but we haven't gotten there yet.
Okay.
There's a whole nother prophase, metaphase in here. I'm just showing you the big picture right now. They will. So, by definition, during meiosis 1, homologous pairs are separated into 2 new cells. One more question. Is this a diploid or a haploid cell? It is diploid because you have a mom copy and a dad copy. Yes? The 2 new daughter cells after meiosis 1, is this a diploid or a haploid cell? Haploid. Did everybody see that? You only have a dad copy and you only have a mom copy up there, right? Yeah? Okay, one last thing has to occur. Now, that's great. So I've separated my genetic information. Woo, that's wonderful. I'm now haploid. That's great. So upon fertilization, I'll have the same number of chromosomes. However, this is way too much genetic information. I mean, there's double the amount that I need in there. So what should I do with this cell now?
Break off?
Yep. So remember in mitosis, what happened? You just separated your 2 sister chromatids. Same thing's going to happen here because you just don't want that much DNA, right? You don't need it. Same thing's going to happen up there. So meiosis 2 is an actual separation of sister chromatids, just like mitosis. Same exact process right here. So I started with a diploid, 1 diploid cell. I end with how many cells? 4, and what are they? Haploid cells. See? Everybody see that? It's like way too much information, I know. You may never get it. But I'm going to try at least. Do you see that? I mean, you're reading it in your book. It says, in meiosis, 1 cell, 1 homologous diploid cell eventually produces 4 haploid cells for gamete cells. And you guys read it every day, but you don't know what it's talking about, right? You're like, I have no idea what that means, but okay. And you might just have to memorize it, that's fine. But I just want to prove to you that it actually happens. One diploid cell ends up with 4 haploid cells. These are now your eggs and your sperm cells, right? Now, this diagram's a little off because right here, it looks like I have the same exact genetic information as here, right? Remember, we're just looking at the big picture. It's actually not. There are several places where there's some recombination going on. This is the same picture I just showed you. So the stages of meiosis are actually interphase, and it goes through all these stages twice. So you have prophase 1, metaphase 1, anaphase 1, telophase 1. Meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2. First division separates homologous pairs. You go from diploid to haploid. You guys told me that. Second division separates sister chromatids. I am still haploid. I just separated that genetic information, right? Just like mitosis. All right, so I'm only going to point out the differences now because everything else is the same. Interphase is the same. Prophase 1. Remember the chromosomes start to coil up? That's great. Same thing happens. But there is something different going on here than happens in mitosis. Look at that picture. What do you notice?
That you have the male DNA, the male chromosome, and then the female chromosome?
Yeah, these homologous chromosomes, what are they doing? They're like pairing up. You see right here? They're actually like sharing genetic information. They're switching out some genetic information. Little recombination
How
there.
can they do that if there's 4 different cells? If there's 4 different chromosomes?
It's not, it's still one cell right now.
Oh, okay.
It's still one cell.
Okay.
Yeah, we started over again.
Okay.
The other one was just the big picture. We're still one complete cell right now.
Okay.
Right? All 46 chromosomes are in here. I'm just focusing on these. And the only difference is now all 46 of those chromosomes, they have gone and found their partners. So the 1s have attached, the 2, just like that karyotype. The 3, chromosome 3, chromosome 7 have found each other. They actually come together and overlap in prophase and share genetic information. Now I can tell you how that happens, but we'll be here all day. Just believe me that it happens. Okay? They switch out genes. This crossing over in prophase 1 is very important. It does not happen in mitosis. And again, it allows us to have genetic variation. So that's why all of our brothers and sisters don't look exactly like us. Very important. When you have the 2 homologous pairs that actually come together, that's called a tetrad, which means 4. That makes sense. And the actual location where the genetic information is crossing over is called chiasmata. Just some new vocabulary words. Metaphase, same thing, lines up at the middle, except that instead of the sister chromatids lining up in the middle, the homologous pairs actually line up in the middle. I'll be really nice about the questions that I ask on this. I know there's a lot of anaphase, I have a separation of homologous chromosomes, right? So I'm just separating the big number again. And here are my 2 cells. Now notice, technically these are haploid cells, but notice that the genetic information is now showing some different colors here, right? Because I've actually exchanged some information. And then prophase 2, metaphase 2, anaphase 2, separation of sister chromatids. It's all exactly the same as mitosis. But you end up with 4. They are haploid cells because their original, where they originally came from, were the same. But again, there's some recombination there, right? So you have some variations of that genetic code.
And it's just going to consistently be doing that so it can form the baby.
Well, so it can form gamete cells. I mean, no. Sperm cells, yes. I mean, guys they produce millions of sperm every 5 minutes. So yeah, that's happening rapidly. And women, no, you're born with, your gamete cells are actually predetermined, and you're born with a certain amount of eggs. So it doesn't happen. Meiosis 1. So here's your prophase, metaphase, anaphase. Look, and here's your homologous pairs. They're pairing up. They're attaching to each other. They're exchanging genetic information. Homologous pairs in the middle. Homologous pairs separating. Notice it's still like that double sister chromatid right there, right? They haven't separated yet. Meiosis 2, lining up. Anaphase 2, separation of your sister chromatids. Telophase, you end up with 4 haploid cells, right? Small amount of information. This diagram is nice if you're kind of comparing mitosis and meiosis, I'll just point out the big picture. So obviously over here in meiosis, you have the pairing. So we start with the same cell, right? But the homologous pairs actually pair up. At metaphase, sister chromatids line up, but at metaphase 1, homologous pairs line up. Anaphase and telophase, again, exactly the same for the separating of your sister chromatids. You end up with diploid cells here because I have male and female copy of every one of my chromosomes. Same exact genetic information that I started with. My haploid cells, they're haploid because they only have a mom copy or a dad copy. Compared to the normal one, you can see they only have one copy, and there's a little genetic variation going on there. So there's a lot going on in these two processes. Just really try to focus on the questions that we go over and your online homework. I promise I won't sway too far from any of that information. In high school, we usually take 2 weeks to cover this. So here's a nice diagram that just kind of shows you the main differences. Mitosis is 1 division, meiosis is 2. Identical mitosis, different from the parent in meiosis. You make 2 cells and they're diploid. In meiosis, you make 4 cells and they end up being haploid. In mitosis, growth and repair, meiosis only for gametes. There's no crossing over in mitosis. And in meiosis, you have this crossing over in that prophase 1, which allows for some recombinations there. During metaphase 1, so we're talking about metaphase 1, right? Of meiosis. I know it's meiosis because it says 1 behind it. Crossing over occurs. Homologous chromosomes line up in the middle. The nuclear envelope breaks up. Centrosomes migrate, or sister chromatids separate and migrate to opposite poles.
1, A.
A? No. B.
Oh.
Homologous chromosomes line up in the middle. So when does A occur, crossing over?
During interphase.
Nope.
Prophase.
Prophase what?
1.
1. Yes? Not prophase 2. Just prophase 1. Very first one. And then let's just look at this one. Sister chromatids separate and migrate to opposite sides. If I'm talking about meiosis, this actually occurs in what? Anaphase 2. Yes? Anaphase 1 would be separation of homologous chromosomes. This is referring to anaphase 2. Upon completion of telophase 1, not 2, 1, and cytokinesis, there are how many cells? 2. And what are they? They are haploid, right? You told me that by looking at that diagram. They're haploid after the first division. Crossing over, very important during prophase 1, as we said, allows for some genetic recombination there. And it's the swapping of pieces of chromosome between homologous pairs, right? Same chromosome. 3 steps, we break, we refuse the DNA. It's much more complicated than that. Which of the following is a characteristic seen in prophase 1 that does not occur in prophase 2? So chromosomes move to the middle, spindle formation occurs, chromosomes have been duplicated, crossing over occurs, or cytokinesis occurs. You see it in prophase 1, but not in prophase 2. There is. Huh? D? B? No. No. Crossing over.
Crossing over.
Right? Guys, crossing over is so important. I'm trying to point out to you that I'm not going to ask you some, crazy question. Crossing over only occurs in prophase 1. Very important. If I'm going to ask you the differences between those processes, I'm going to stick to the big differences, right? So crossing over occurs in prophase 1, does not occur in prophase 2 of meiosis. Does it occur in mitosis at all?
No.
No. So we talked about this crossing over that allows for some recombination. There's another factor here of why you don't look exactly like your siblings, unless of course you're identical twins with any 2 parents. A zygote with over 70 trillion different combinations, right? So, you've got, it just depends on which sperm actually gets in. But, couple 1 can have random having variations here. So the point is that you're not making clones of yourself, right? You allow for some variation there. Thank the Lord. Sometimes it doesn't always work out perfect. Here's a great example. In meiosis 1, these homologous chromosomes didn't separate. Whoops, right? So now you have an extra copy over here, and you end up with some zygotes with extra copies of chromosomes. That's called non-disjunction. Non-disjunction can also happen here in meiosis 2. You don't have a separation of sister chromatids. So what happens? You have an extra copy over here. Guys, the ones that have are missing chromosomes, or they only have one copy, they usually just like fuse back into the body. Like the body, again, it's called apoptosis. Your body just eats it and uses it as fuel because it's so messed up. But ones with extra chromosomes, sometimes the body's like, oh, okay, I can go with this upon fertilization. And a good example here is Down syndrome, right? Trisomy 21. You can actually see he actually has an extra copy of a chromosome there. So, you know, a lot of times, again, sometimes upon fertilization, I don't know statistics, but oftentimes like miscarriages or early miscarriage pregnancies are due to the fact that something may not have been right with the egg or the sperm, right? And so that's just part of nature. It just kind of happens. Of course, sometimes, you know, I used to live next door to this poor lady, bless her heart. She tried to get pregnant for like 20 years. And they finally did. And then late in her pregnancy, they started to notice like there were like major deformations with the baby. Obviously, that zygote or that baby probably should have not survived, but it did sometimes against odds. And so, you know, but the cell, its natural cycle is to not allow those things to occur, right? They have checkpoints. So the cells actually, they fuse with lysozymes and they just integrate themselves back into your body as part of a food source.
Now,
yes.
the deformations, or then that occurs during this process, correct? Like
No, there are 500 million reasons that could cause a deformation in a baby. This is just an example of the cell cycle.
I was just wondering because there are some babies who and I know this is rare, but sometimes the brain is born on the outside, not necessarily inside the skull. And I've heard of that, or where some vital organs are not necessarily growing in the right places.
Absolutely, yeah. No, there are, and those are probably, now, there's all kinds. This is just an example of something that may happen from an irregular cell cycle. There are 50 million things that can happen because the embryo doesn't develop properly for some reason. There's genetic mutations. There's environmental conditions. I mean, if you live anywhere near a nuclear power plant, your kid may have an extra finger. Like, there's all kind of just a plethora, which is why we can't get our hands on the cause of things like, we can't fix any of these things because there's so many factors. This is just for cell cycle, but you're right, there's a plethora of reasons why a fetus may not be born, or they may be born early, they may not be born at all, they may be born with, you know, some type of autism. Well, we don't even know what causes that. We still don't know what happens with that. So there's so many unknowns, but this one is pretty well studied. Down syndrome is pretty well studied, but this is just an example of a cell cycle that didn't properly separate either those homologous chromosomes or those sister chromatids. One difference between mitosis and meiosis is mitosis produces cells genetically identical, but meiosis does not. Mitosis produces more daughter cells. No. Meiosis is needed for growth. No. Mitosis produces haploid cells, but meiosis produces diploid cells. Opposite is true. That's not a false statement. Mitosis requires only one parent cell, but meiosis requires two parent cells.
Yeah.
Fertilization requires two cells, but not meiosis itself. So the best answer is what? A. Mitosis makes genetically identical, and meiosis does not, right? Has some genetic variation there. Do that crossing over. Crossing over during prophase 1 results in non-disjunction, translocation, reciprocal translocation, duplication, or genetic recombination. E. Genetic recombination. Okie doke. Is this we are running not very fast on time. We're just going to answer these two challenge questions. And the second challenge question, this one you may answer outside of class. optional points, and turn it in. Either you can email me the answer, or you can bring it to me on Monday. But this one I'm not collecting in class. Everybody hear me say that? But you can still answer it and turn it in later. Okay?
That was the second one.
The second one, the one with this one.
That was the one I was stuck on.
Yes, this one, and I can answer questions about it too during class. Right now, I want you to answer these two on a post-it for in-class optional points. Victoria?
Do you mean if you want to say because you're not going to be
I won't be here. You can either email it to me, or if you want, the best thing to do with that question is put it in a Word document and submit it online, because I could lose. Yeah. It's best for you to submit it online, because I lose stuff. All right, do 1 and 2. And then we have probably about 30 minutes if you want to finish up your homework and or have any additional questions, and then we'll move over to the lab, okay, for lab number 12. And in the meantime, I'm going to give back your Post-its. I have quite a few labs to get back to you as well, and if any of you have